Alex is from Buffalo. I am a neuroimaging researcher by trade, so I'm not necessarily an ontologist. I'm a neuroscience PhD. So a lot of what I'm going to talk about is kind of driven by pragmatism, my desire to use ontologies in my own research projects. So I just wanted you to keep that in mind. That's kind of the angle that I'm approaching all of this from. So I'll go through a little bit of the background, sort of the motivation of why I want to use ontology for my research, a little bit of the background in biomedical imaging and neuroimaging specifically, and kind of where ontology fits in. And then from there, I'm going to take you through kind of a brief overview of a couple of the terms that we developed in my own ontology, which I'll discuss a little bit towards the middle of the talk. And I'll discuss kind of the motivations that we had for why we did some of the terms we have, where they're being used. And then finally, I'm going to wrap up with some of the use cases I found for these terms. Specifically, it's being used in my PhD to power a neuroinformatics platform. And I'll walk through each of the places where that gets used. Okay, so to begin with, I'll just go through the brief introduction. So biomedical imaging, for those unaware, it's a method of visualizing the internal anatomy and function of some subject. Doesn't have to be a person, but I typically work with human subjects. It's been a mainstay of modern medicine for decades. Includes things like PET CT, MRI, X-ray, any kind of biomedical imaging. And so it typically looks like this little cartoon over here that I like a lot. It's a little morbid, kind of showing a person getting cut up like a ham. But it does a nice job of illustrating the way that we work with these scans that we use in biomedical imaging. So you can see that there's these, instead of two-dimensional pixels, we use these three-dimensional voxels. And so that's used for getting a nice look at what a person looks like or a subject would look like in three-dimensional space. And so I'm going to talk a little bit throughout this presentation about clinical imaging and research quality imaging. And so the reason that I'm going to do that is because there's quite a bit of difference in how imaging is done in a clinical routine. So this is things like doctor's visits, and in the research setting, which is where I typically work. So in the clinical routine, you have usually faster, lower quality images. You have a patient come in for some scan that they need to get done for diagnosis or treatment. And so you usually want that to be pretty fast. You just want them in and out, and you want to treat the patient after that. But this happens for pretty much anyone that comes in for any sort of imaging. So you have a ton of data lying around in that. It's not really stored in any way that's easy for researchers to access. And on the flip side, we have the research or the research setting, which typically requires longer and higher quality images. It's used to find something specific out about the body. So you need quite a fine level of detail. So there's a bit less data in there. That's not to say there aren't large data sets in the research setting. There are plenty, and I've worked with them. And I'll talk a little bit about them later on. But really, there's a ton of data that's not being used in the clinical setting. And so a part of why this is important is because imaging is very expensive. So when we're doing imaging in the research setting, you usually have to write in a lot of money in your grant to be able to acquire new images to work with. So it's very useful from a neuroimaging researcher's perspective to be able to reuse data. It's a very powerful thing, just beyond all the FAIR principles and everything that we've been talking about all week here. So there's been a big push recently to be able to mobilize a lot of this data and allow us to use it in our research setting. And that in turn would also allow us to empower clinicians and clinical researchers to also get more out of their clinical biomedical imaging data. And so you can't really talk about biomedical imaging without talking about DICOM. So DICOM is it's recognized by ISO as the standard format for storing and transmitting biomedical images. Any sort of biomedical imaging modality, PET, CT, MRI, they will all use DICOM. So there's a lot of interoperability in there. And what a lot of the interoperability comes from is the standard use of tags and DICOM headers, which encode a lot of pertinent information about how the image was acquired, where it was acquired, some information about the subject being scanned. And because it's such a ubiquitous technology, there is a huge ecosystem of software and tools that make use of this DICOM standard. So it's very easy to work with interoperably. And then more specifically in the realm of neuroimaging, we have what is called the BIDS, or the Brain Image Dataset Specification. This is much newer than the DICOM. It's not, it doesn't have quite as much adoption. But there's been a push in recent years to make more use of this BID specification. It's not necessarily ontologically sound. It's kind of just a prescribed format of laying out your images on a file system. But it is great for reproducibility and interoperability because it specifies how all of your images have to be named and placed in a standard format that you can just reuse over and over again. And they provide different kinds of programs called BIDS apps that will just take in a BIDS data set, and the user doesn't really have to do anything with it, and you can just start to use it. So helps lower the barrier of entry for people getting into neuroimaging. And that'll bring me to where ontology fits in with all this imaging data. So much of the data encoded in DICOM headers is semantic strings. They often differ from institutions and manufacturers, so it's very messy to work with. You think you have your standard that you use to make your data interoperable, and it's really not like that in practice. The data can be very different from one data set to another. This makes a high barrier of entry for working with imaging data, so you have a lot of clinical scientists that will want to use biomedical imaging in their research, and they don't really know how to. They would have to come to someone like me at my lab and say, okay, I need you to do this for this study. And it's a lot of effort involved with that. So biomedical imaging data may be represented with these high-level classes that integrate with DICOM and BIDS, and this will help harmonize the large amounts of existing data. And so now that we have our use case for our ontology in biomedical imaging, I want to discuss a little bit some of the terms that we have posed to help us tackle this. So all of this is coming from the work that was started a few years ago and presented by Dr. Sarah and Dr. Deal in collaboration with my PI, Dr. Dwyer, at ICBO in 2019. So this all comes from MRIO, the MRI Acquisition and Analysis Ontology. So it was constructed in a top-down and bottom-up development using OBO Foundry principles, extending OB and IAO. There's about 70 new base terms to represent the way that you acquire and analyze MRI data. And so building off of that, I kind of took a lot of the work that was done with that and kind of refactored it out into something a little bit more modular. When we had first started working on MRIO, we were using the output of an imaging assay to be image, which is how it's defined in OB. And one thing that we ran into with that, and I'll talk about this, I think, in another slide, is the way that image is defined is it's specifically a two-dimensional object. And I showed you the nice ham person earlier on in the talk. We typically use voxels, or 3 dimensions, and sometimes 4 dimensions or even more, depending on the modality, to represent imaging data. So we kind of needed our own term to fully capture what we're working with. So we're posing this image data set class, and we're contributing it to OB. There's an open PR that I have to take care of to finish merging it, but we've worked with the OB team. And we're defining it as a data set that is comprised of structured measurements about some entity and its associated metadata using pixels, voxels, or an arbitrary number of dimensions. And then we have a gloss saying an image data set can be the source from which an image is produced. So it's just a nice collection of the imaging data that we would get from our biomedical imaging, biomedical images, as well as all the other data that's associated with it in the file headers. And then for working with the data, we also posed a new class called image dataset analysis, which we're defining as the process of defining a data item from an image dataset using computer algorithms. The produced data item can be an image dataset, data measurement, or any other data item. And this is going back to what I was saying with why are we coming up with our own term when something like image already exists? And like I said, image is specifically defined as two-dimensional. Specifically, it is a subclass of figure, which really emphasizes the importance of how you arrange a two-dimensional representation of some sort of ICE. And like I said, that doesn't really fit with how we work with our biomedical imaging data. So that's we needed a more general term for an information content entity that would fully represent the kind of data that we are working with. And we thought that dataset is a very fitting parent class for that. So we made a new class and we called it image dataset. And so the hope with this is I'm using it mostly in neuroimaging to work with MRI, right? But the hope is that since the DICOM standard is used for almost all biomedical imaging modalities, and we've kind of geared this towards working with DICOM, that you can extend this image data set to be used with really any other kind of biomedical imaging data. So you don't necessarily need to have what we call the MRI image data sets. We'd have a PET image data set, a CT image data set. Also, the flexibility in end dimensions makes it a little bit easier to work with different kinds of imaging data or other kinds of imaging file formats that will also encode information inside the file itself. And so this is kind of an example of how we've been using it and how we tie the DICOM standard into it. So on the left, I'm not going to belabor the details. If you're not in neuroimaging, you don't care about this, but these are just different kinds of MRI that we can acquire, and they're all acquired in different ways depending on the machine settings that get encoded into the DICOM headers. So this is how we're tying the DICOM standard into our image data sets. These are just some of the DICOM headers that you'll find. There are many, many more. This is just a simple representation of what our imaging assay would look like. So we had started this a couple of years ago before there was an MRI imaging assay in OBI, which we are now using. So we've updated that. But you see that we have our imaging assays that will produce different kinds of imaging data. And we relate it back to a participant and also produce this image dataset class. And then for the analysis, we will take the image dataset as an input. We'll do any kind of analysis that we have encoded in our ontology that produces some sort of IC out of that. And I'm going to wrap up by talking about how we're using this in my project. So I personally think that this is the more excited part, but that's because I've devoted 3 years of my life to building this. But I'll go through and show you how each part fits into the platform. So this is kind of a broad overview of how it works. So we have these unsorted DICOMs, unannotated images coming in. And in order to work with neuroimaging data, you need to know what kind of image you're working with. So we tag them. We tag the DICOMs as they come in. We do some automatic classification to say what it is. We then pass it off to a Sparkle query that will say, okay, we have this kind of image. We can do these kinds of analyses with it, and this is the kind of data that we get out of it. And then from there, we can pass it off to a database that stores all of that. And then we can do our queries on that using the OnTop software, which I'll talk about in a couple of slides, to provide clinicians and clinical researchers the ability to work with this data, access it, generate new hypotheses. So here's the kind of the ontological representation of how we do our scan classification. So this is just an example of what an individual scan would look like. So we have it represented here with some of the terms that we've added and how they tie into DICOM. We can run the Hermit reasoner and it'll say, okay, this is the kind of image that we have. So we've been doing this for a good amount of time, but it's a little slow, wasn't super accurate. So we actually switched to using XGBoost, which is a gradient boosted decision tree algorithm, which is much faster. But the point of that is that when we were training it, we used these terms that we had encoded already in our classes, so it's kind of used to inform the machine learning model. So it's not a direct integration, but it was pretty close. And XGBoost gives you a nice overview at the end after you train the model of what it thinks the most important features are. And after reading a DICOM header and training it, it did say that these were the most important features. So our domain knowledge that we had encoded in our ontology lined up with what the machine learning model found. And so the next step after that, once we know what images we're working with, is to assign the analyses to them. So over here, I have just an example of a function that Mackenzie Smith, who was a student of mine who graduated this past year, developed that would take in any sort of labels that you have from the ontology, and it does some string processing to match them up to the correct identifier. It'll go through and find any analyses that will use these input images in any sort of combination. So it does a bunch of permutations to find it. Right now, it's just some simple hashing between the labels and the identifiers, but there's potential there for some more robust querying systems. If anyone is good with NLP, I've personally never done it, so don't know much about it. And then finally, the ability to query data out of this platform was probably the most important part of us. Is something to deliver a tool to clinical scientists. So we use the OnTop software, which will take some mappings from your RDF triples to an SQL schema or some sort of relational database schema. We use Postgres for ours. And it'll create what's called a virtual knowledge graph that it can then query over and tie it into the data, depending on how you've annotated based on your ontology. And then the database is something called XNAT, which is something from the neuroimaging world. It's a pretty popular way of storing your neuroimaging data. And it's got its own schemas that they've built up and used. So I couldn't really make any database schemas based on my ontology. I had to work with what XNAT said was the schema. So I needed OnTop to go between the ontology and the schema. And so this is just an example of something that we can do with it. We have our Sparkle query that just says, find me everything in the database about the thalamus. So say a clinical researcher comes in and says they want to know something about the thalamus. And so we'll return something like this thalamic volume measurement datum, which is a term that we've come up with, said is about the thalamus, which we pull in from Uberon. We also pull in stuff from Mondo to do some clinical queries. And so because we have it all kind of tied together nicely, this simple query can pull out something such as this thalamic volume measurement data or any other data that we have annotated as being about the thalamus. And so this is kind of like a cheap prototype-y view of it, but this is how it looks in practice currently. So a clinical researcher can go in and say, okay, just find me everything about the thalamus, and it'll plot these distributions of the results that we have of our analyses that were based on the thalamus. So we have just some measures of thalamic volume, like I mentioned in the previous slide, as well as a full data set that they can then download and work with however they see fit. So if they want to use this to generate some new hypotheses, or if they just want to test something about the thalamus, the data is all there. It's nice and easily contextualized for them to work with. And then finally, this isn't necessarily being used for my project, but I think it's an important step in neuroimaging research, is the integration with the brain imaging data structure, or BIDS. So on the right, this is, I was talking about it earlier. This is an example of what a BIDS data set would look like. So as you can see, it's all named pretty consistently. The names are just the different kinds of images that you work with. So that's related to how we define the acquisition of our images in MRIO with our image data sets. So you can kind of take a similar approach to what we were just doing, where we were automatically classifying the scans and then moving it into this standardized data set to make it easier for people to use and reuse their data. This is an example of what the transformation would look like. So when you have your DICOM images, they're pretty unintelligible to look at them, just based on these names and the way that the files are called and the way they're organized. And then over here, you have your nice, organized, easily digestible BIDS data set. And so in conclusion, we believe that our work using the image data set and image data set analysis class helps harmonize large data sets, helps harmonize working with large data sets in biomedical imaging. In particular, we show that automated scan classification and assignment of analyses makes it easier to work with large unsorted MRI data sets. And additionally, integration with DICOM and BIDS helps clinical and translational researchers leverage their existing neuroimaging data. And as I've been saying throughout this entire talk, most of the development has been focused on MRI, but we believe that image data set is a suitably high-level term that may be used with other imaging modalities. And then I just wanted to highlight down here, there is a typo here. I wanted to highlight that. So we're working with integrating this with OBI. There's been some discussion of renaming image data set to imaging data set, so that should say imaging data set. So the name might change if anyone ever decides to work with MRIO at some point, or it's in OBI and you see it in there. it's a different name, that would be why. And I just want to wrap up by acknowledging the team that I've worked with from Buffalo, in particular Lucas Serra, who did a lot of the groundwork for MRIO, Alex Steele, my committee member, Bill Duncan, who have taught me most of what I know about ontology, and then my PI, Michael Dwyer, who's taught me everything I know about neuroimaging. So, thank you.
So we take one or two questions. Please.
So the DICOM standard is very extensive, right, and very good and very. So in that standard, there are lots of things that are described at the image level or at the modality level, right, like MRI or things like that. So how strictly have you adhered to the DICOM standard in modeling out this ontology, right? As an example, slice thickness, right? That's something that is relevant to CT, but also to MR. Put it too deep into the MR one, then the CT one can't use it. What are your design principles there?
Yeah, so for those on Zoom who might not have heard the first part of the question before we give him the mic, he was asking about the hierarchies that are in the DICOM standard. So you have hierarchies of your modality specific tags, your image specific tags, series studies, things like that. And he was asking how we tie that into our ontology. And I'd say we tried to abstract that away from the DICOM standard a little bit. We tried to have more of a general representation of how it works. Mostly, like I said, this is mostly an MRI. So it might be the case that we would need to refactor something like you said, for example, slice thickness, which might not be entirely interoperable between modalities. So there might be a need to kind of differentiate things with some terms that way. But I'll go back to the hierarchy I have here. So it's, as you can see, it's mostly tied under these MRI specific things. So like I said, we're trying to keep it general, but it's also because it's use case driven and an application ontology thus far, it is very specific to MRI. So that is a very good point.
Okay, I think we should move on. Thank you very much, Alex.
Thank you.
Good morning, everyone. I'm Pradeep Dekoda. I'm currently pursuing my master's degree in computer science at the University of North Carolina, Greensboro. And today I'll be talking about knowledge of the ancestors, intelligent ontology-aware annotation for biological literature using semantic similarity. I'll briefly be talking about the consumption of Go ontology here to automate the annotation of scientific literatures. Also briefly talk about the steps that's generally taken in automated annotation. I'll also be talking about data pre-processing and model training, and finally the results or the performance of the model itself. So this is a part of a scientific literature, and the highlighted words here are some of the ontological terms. So our approach here is to make the model learn from these and try to see if it's actually possible to annotate these terms using machine learning or deep learning in general. So previously, for automated text curation, there was approaches like syntactic analysis, lexical analysis, and traditional machine learning to do all of these annotations. They were generally not efficient or not accurate in the sense that most of them were rule driven, and they were not able to annotate concepts that they had not seen in the training data. So in that regards, deep learning has started to perform better, and that's why you would see that most of the researchers these days are using deep learning for these kind of tasks. Here are some of the recent work that I have gone through, and they are pretty much using deep learnings. And to briefly talk about these, generally automated text curation is considered as two-step task, where the first task is to decide whether a given word is an ontological concept or not. And then the next task would be to assign a particular concept to that word. So, in this slide, I'll be talking about the limitation of the annotations or the general way of doing things and how it limits the model from being aware of those ontologies. So as you can see over here, we have a word mitochondrial, and the ground truth here is that it's associated with the Go ontology 5739. But then say our model fails to predict that, and say it predicts Go.70, which is not ontologically related or semantically similar. So in that case, what we want the model to do is, if it fails to predict the exact term, we would want the model to at least predict some other term that is semantically similar or semantically closer to our ground truth. So as I said, the goal here to develop a deep learning model that is ontologically aware in recognizing these texts. So for the training purpose, we have used Craft, which at this point is pretty much a gold standard for automated text curation tasks. It has around 29,000 sentences and pretty much covers around 25,000 concept annotation related to Go ontologies. So that's the reason why we decided to go with this data set. And this is the general pipeline that we have considered for our deep learning model training and testing. So we have the Craft annotation and articles, and for, in that, since it's a big article, we have to parse it to break it down in sentence level, and then we do some annotation handling. We divide the data set into train and split test, where some of the data are kept for training and some of the data is kept for testing purposes. And on those data sets, we do the pre-processing like tokenization, where now a sentence is further splitted into words, and there's a mapping, a word-to-word mapping between the words and its correspondence with an ontological term. It could be an ontological term, Go ontological term, and in that we would say that it's Go certain terms, or if it's not an ontological term, we say that it's O or out of concept annotation. We'll then do IOB tagging and concept annotation, which I'll talk briefly in the next slides. We have also augmented the parts of speech information into that, so that it makes the model more aware of the information and helps understand the model more. So after all of these processes are done, we have our pre-processed training and evaluation data set. The pre-processed training data set is used to train the model, whereas the evaluation data set would then be used to evaluate the performance of the model. Talking about the IOB format, it's one of the common format that is used for IOB tagging. It's also part of span detection, where we detect whether a term is a Go concept or not. So if it's if it's not a Go concept, we say it's an O, so representing outside of a concept or not a concept. And if it's a Go concept, if it's a phrase, then the first word is assigned a B term, and then the rest of the word are assigned items. B meaning beginning of the concept, and I the insides of the concept. So like I said before, automated text curation is generally considered as two steps: span detection and concept normalization, but we have merged them together to say that if it's a beginning of a concept and it is annotated to Go X, then we say that instead of dividing them into two different tasks, we simply say that it's B dash Go X term. Now, talking about the pre-processing steps, we have used Craft version 4.0, and with the introduction of version 4.0, there were some complexities, complex annotation formats that were introduced, and because of that, we have to do quite some pre-processing in that. And so, in talking about the pre-processing, let's say if there are no annotations, then it's pretty much a straight process. We break the sentence down into token level, where for each token, there is a one-to-one mapping between the tokens and the tags that we're supposed, or the concept that we're supposed to predict. So as you can see, if there are no annotations, then they all belong to O tags, meaning none of them are in the concept, so they are outside of the concept, all are O's. But in this kind of case where there are actual ontology terms, then we again split the sentence into tokens, and for each of the tokens we map each of the tokens with a Go term. So here pedicles would be associated with B-Go 44316 and so on. Here is a complex situation where we have overlapping annotations. So in this case, the vesicle formation as a single phrase is annotated to a Go 6900, but then within that phrase, we have a subphrase vesicle, which is annotated to another Go term. So in this instance, what we have actually done is we have broken down the sentence. We have made copies of that sentence into two different sentences, where in one of them we are just considering vesicle or the subphrase and removing the phrase, whereas in the next phrase in the next sentence we are keeping the entire phrase and annotating it to a different term. So in the first case we have since we have only vesicle, it is annotated to 31982, but in the second case, since we are considering the whole phrase, it is annotated to a different Go term, B-Go and then I-Go. We also have something called discontinuous annotation, where the annotation here is discontinuous. It's it's B/P formation, but instead of considering the entire span as a single annotation, it's partially ignoring the phrase in the middle. So in this case, we had to make the model more less confusing, and to remove this uncertainty, we have removed that part of the sentence and then treat it as a regular sequence. Now, for the actual deep learning model, there is we have something called embeddings, and we have our actual learning algorithms. So to talk about embeddings, our deep learning model would not understand the words that we use in our articles. It only understands numbers. So there has to be a way to convert the words into numbers. So how that is done is we associate each of the unique words in the corpus with a number, and then we let's say if there are 3,000 unique words in the article in our training corpus, then each of the word is assigned a 30,000 dimensional sequence, which is if you think about it, it is computationally very expensive for a model to treat each word in a 300 dimension, and then to consider the entire corpus. It's computationally very expensive. So what's generally done is that during training, the higher dimensional words are projected into a smaller dimension. So in this case, say we can reduce it down to 100 dimension or 300 dimension. So craft here is the embeddings that we have trained, that we will train as the model is trained on the core training data set. The embeddings will be learned in case of craft. In case of Glob and Elmo, they are already pre-trained. Glob is trained on 2 million corpus, and Elmo is trained on 800 million corpus. So these are already pre-trained corpus embeddings, and they give a pretty good latent representation of the words that are present in our corpus. For the deep learning algorithm, we have considered gated recurrent unit in this case. Gated recurrent unit because it considers it can utilize the fact that we are not just dealing with token level tokens, but sequence as a whole. Since it can understand a sequence, a lengthy sequence, that's why we have gone with GRU or gated recurrent unit. We have also made a comparison with BERT, bidirectional encoder representation from transformers. So this is the model architecture. The graph was not supposed to look like this, but to briefly talk about it, this is the snapshot of our model architecture. The lower part is you can consider them as the inputs in a given timestamp. So we are dealing with these 3 tokens for simplicity. For each of the words, it passes through pre-processing, as I have talked before. So each word is tokenized, it's associated with its parts of speech. And then we also have something called character representation, where a token is broken down further down into as a sequence of characters. Then they pass through embedding layer that I briefly talked about in the previous slide. We concatenate these inputs, which is again passed through a bidirectional GRU unit. This combines the inputs from previous layers, and finally we have dense connected layers that makes all the association, and then we are able to predict the final term as such. So to talk about how the output looks like, so for the output, it's not a general, we cannot say that, or other model cannot say that given a token, it's associated with that particular Go term. It is actually a probability distribution where it assigns the highest probability to one certain term and then certain lower probability to other terms. So in the training, we say that if we are dealing with this term, we say that we'll provide the highest weight to this term, and then the lowest weight, in this case, 0 to other terms. And this is what we ideally the models would try to predict, or try to make, will try to train the weights so that they are closer to this kind of numbers. But what happens in reality is it tries to make it close to that, but it cannot get absolutely exactly similar to that. So in this case, we have something as this, which is also a probability distribution, and then we say that because this index has the highest probability, the term must be associated with B-Go226. And in the next one, we say that because this is the one with the highest probability, it must be associated with 6916. So this is how our prediction actually works. So how our approach is different in this sense is that rather than treating a token as a single token as related to a single Go concept, and then ignoring the fact that they might be related to other, or they might be semantically similar to other Go terms, which is the general case. As you can see over here, we say that provide the highest weight to one of the Go term, and say that other terms are totally independent of each other. But in reality, we see that if we take a look at this graph, we can say that if we are trying to predict, if we are trying to work with Go 226, then Go 7010 or 6996, they are not totally independent of each other. They are semantically similar to what the model should be predicting. So in this case, we say that, yes, since we are predicting 226, provide the maximum weight or maximum value to that particular term, but also consider the hierarchy of the Go ontologies and associate some weights to other terms as well. So if we do that, the model during training will be aware of the hierarchy, and then if the model actually fails to predict the exact Go term, it will try to make a closer prediction to something that's semantically similar to that term. Now talking about the performance evaluation, we have considered precision recall and F1 score. We are more interested in F1 score and Jaccard semantic similarity here. Jaccard semantic similarity is what we have used to make that association and to make the model understand the hierarchy. So this is what the performance of the model looks like. We have the baseline model, which is trained on different embeddings, as I have explained before. As you can see, the F1 score and the Jaccard semantic score for something like craft and gloves are relatively lower than the Elmo. In the baseline model, Elmo appears to work best. And then we have our ontology-aware models, again trained on craft, glove, and Elmo. And as you can see, F1 score of Elmo is highest, and so is the Jaccard semantic similarity. And if you look at if you compare the F1 score and the Jaccard semantic similarity here, since the Jagged semantic similarity is higher than the F1 score, we can say that in cases where the model has failed to predict the exact bottom, it is somewhat capable of predicting semantically similar or semantically close terms. We have also made a comparison with BERT, which again appears that it's performing better than BERT in this case. For the future work, we would like to augment the association with some biological sources like Biothesaurus and UMLS. This is to make the model more aware or help the model understand better. Also, we know that for model training, the higher the frequency of data, the better the model performs. So in the corpus that we have used, we have some terms that have that occur in higher frequency, and then some terms that generally they are less frequently occurring. So in that case, we can use the ontologies to use the synonyms of the ontologies, and then bump up the frequency of the lower underrepresented terms. We can also use the concept of boosting, where we are not just associating the probability of one particular core term, but then we are combining or we are considering its subsumers or subsumers probability into consideration, so that we can boost up the entire score. And the data that we have used, as I explained, is version 4.0, which is publicly available at this link. And then the source code is also the source code that we have used to generate the result is also publicly available in GitHub. The source code is also archived in Zenodo at this DOI. Finally, I'd like to acknowledge that this work is funded by a career grant to Dr. Prasanthi Manda from the Division of Biological Infrastructure at NSF. Thank you.
Thank you. That was an excellent overview of your work. And you're in very good company with a lot of good established tools, such as Apache CTAKes or CLAMP coming out of Texas, as well as even Amazon Medical Comprehend. Have you thought of comparing your tool, and what would it take to compare?
That's a good question. To make the comparison, I would say that the comparison here in terms of model performance, we have used a different matrix. So the numbers which we displayed are after removing the commonly occurring terms, or since there are many terms, out of concept terms that are associated with the entire corpus training, it makes the score as generally biased towards those terms. So what we have done is to remove the biasness. We have removed the frequencies or the numbers that have the matching outside concept terms, and then we have our results. So in terms of your question, I would say that to make that comparison, we would need the same kind of benchmarking, and then I guess if we do that, we can make that comparison more accurate.
Thank you very much. No more questions. I guess we'll move on.
To be here today, especially because this has been occasion to see a lot of friends that I've been seeing for a long time. So I am working at the National Institute of Environmental Health, and I've been working there the last 2 years. I just joined the NIH 2 years ago. And so, as many of you know me, I always work on the biomedical, strictly biomedical ontology. And so this environmental health is kind of show me a different aspect on how an ontology can help. So today I'm mostly I would like to let you know about this effort that is going on inside NIHS with hope that a lot of you will join and will help us. And in particular, the last part of the talk will be about one initial use case we have started working on. So first of all, what is an environmental health? This is kind of, as you can see, a very wide kind of definition, where environmental health is assigned to study the effect of exposure to environmental factor on human health. And so these environmental factors are really could be from several domains and things. And so that's why it's very complicated to be able to have a language that can be useful for all the different domains. As here are the different kind of, if it's working, the different type of data types that are filling under the umbrella of environmental health, so data about climate, radiation, food access and quality, water quality and access, financial, infection agents, air quality, chemical activity, and occupational. And I'm sure I left something out. So really is a wide domain that environmental health cover. And as you can imagine, each of these domains has to say their own way to collect the data, to annotate the data with very specific kind of language. And we, the goal that we really have with this initiative is trying to find a way to harmonize the language between all these different domains in a way that we could communicate better the information we are getting from the data, and in particular, we can integrate better information, because to understand